The cell cycle consists of two main phases. You have interphase and you have mitosis. Now, if we were to have like a clock here, right, and one rotation of the clock is the cell's entire life here. So let me put some tick marks on our clock. 1, 2, 1, 2. Right? The cell would spend most of its time in interphase. It would spend 90% of its time in interphase. But it would spend the remaining 10% of its time in mitosis. And at the end of mitosis, we end up with two daughter cells. So if we start out with a cell here, it's going to be a single cell all the way here, but then at the end of mitosis, we have two identical daughter cells. That word identical is usually a good indicator that we're talking about mitosis. Identical daughter cells. Also, we can talk about the number of chromosomes. So you guys now know, because we've done the genetics and all that good stuff, that every person has two copies of each chromosome, ignoring for a moment the sex chromosomes in men, where they have two different chromosomes for that pair. They got one from mom and they got one from dad. These are what we call the homologous chromosomes. Now, in meiosis, we separate the homologous chromosomes, but we do not do that during mitosis. Instead, what we do is we break apart the sister chromatids, these two halves of the X. Sister chromatids. Okay, so we break those in half. And because each daughter cell is going to get one half of this chromosome, and they're going to get one half of this chromosome, they get the full set of genetic material. So if we start with 46 chromosomes, each daughter cell will get 46 chromosomes. When you have two of each set, we call that 2N, or diploid. And in mitosis, you start with a diploid parent cell, and you end with two diploid daughter cells. Right? We break the sister chromatids in half, but they still have 46 chromosomes at the end. So what does a cell look like when it is in interphase? It's going to have all of its chromatin, all of the DNA, and now we've talked a lot about DNA, so you guys should have a good handle on what this is like. All that DNA, it's going to be centered in the nuclear membrane. It's going to be in this nucleus here. And we're going to have a really dense region called the nucleolus, right? So if you see that nucleolus, we're in interphase. That's pretty much a guarantee. There's a kind of organelle in the cell, which we call centrioles. And they kind of provide an anchor for the cytoskeleton, right? They're made out of microtubules, same as most of the fibers of the cytoskeleton, and they just kind of anchor it in place. But they have a special role that comes up during cell division. There's a complex of proteins around them, which we call the centrosome. Plant cells still have a centrosome, but they don't have these little centrioles. So they're just kind of part of the process. Now, interphase is not just one thing, it's actually got three subphases. Just like mitosis has subphases, interphase has subphases. You have the G1 phase, the S, and the G2 phase. I'm running into my own little doodles here. In G1, we've just completed mitosis, so the cell has to get up to its correct size in order to continue doing all of its cellular functions, right? So it's going to have to build up its mass. It's going to have to make more membranes. It's going to have to make sure that it has the right number of all of the organelles floating around inside the cytosol. So gap phase 1, which is what this stands for, is gap phase 1, is a growth phase. We just went from one big cell to two half-sized cells, so now we need to build them back up. We're going to grow. S phase, once we've gotten everything under control and we're back to doing our normal work of being a cell. If we're a stomach cell, we're excreting digestive enzymes, or a skin cell, we're making sure that everything's springy and protecting the interior body cavities from invaders, all that kind of good stuff. Once we're done with that G1 phase, now we are in a good place to start duplicating the DNA. So S stands for the DNA synthesis. Now, you guys have already seen how DNA is replicated, right? It happens during S phase. Before, when we were talking about it, we were just like, oh yeah, the DNA gets, all the chromosomes get duplicated, right? But now you've actually seen the whole process with all those different enzymes of how DNA copies itself. DNA synthesis, DNA replication, is the process where we go from one parent strand of DNA to two daughter strands, right? You go through that whole process, you end up with two daughter strands. What are those daughter strands? They're the two sister chromatids. That's what we're doing during DNA replication. We're making these sister chromatids. Each chromatid has exactly identical genetic information. So when we inevitably break them apart and give one to each daughter cell will get one full copy of that chromosome. Then in G2, we're ramping up for cell division. So we have to build up a lot of ATP. We got to build up more of these organelles in the cytosol. We got to increase the size of the cell to make sure that both daughter cells are going to get a good start. Yeah, we do growth again after cell division, but we do a little bit of it before cell division as well. We're going to just keep growing and growing and growing until we get to kind of a critical mass where the cell has no choice but to divide. So that's interphase, and it always looks like this in interphase. The chromatin is loosey-goosey. It's not bound up. In mitosis, you have, let me see how much space I have here. Yeah, I'm going to make a little bit of room for myself. Okay, so once the cell is ready, it transitions out of interphase. So it did G1, it did S, it did G2, and now it's going to transition into mitosis. Step 1 of mitosis, prophase. You guys might remember that I like to use the analogy that the chromosomes are about to go on vacation. Chromosomes are kept inside of the nuclear envelope throughout the entire course of their lives, right? As soon as the cell divides, we rebuild these nuclear envelopes. Before the cell actually divides, we rebuild some of these nuclear envelopes, and the DNA has to stay inside the nucleus all the time, right? We allow the RNA out, you guys might remember, but the DNA has to stay contained and protected. And the only time it gets to see the rest of the cell is just this once. So they're going on a trip. We have 2 sets of chromosomes here, and I'll see if I can maintain my color ratio. Yeah, I'll do it this way. There we go. So we have 2 sets of chromosomes here. This is a cell that is diploid. 2n equals 4. It has 4 chromosomes total, 2 of each kind, 2 of chromosome 1 and 2 of chromosome 2. 2 of n is the number of pairs. So we have twice the number of chromosomes as we have pairs, meaning there's 2 of each one. First part of prophase, the chromosomes are going to get packed up. You got to pack your bags, right? You got to get yourself together. They condense. Right? And that's why we can actually see them now. That's a dead giveaway that we're in mitosis. In interphase, it's one big blob of chromatin, but in mitosis, you can actually see, oh, look, an individual chromosome. Oh, look, a little leg of a chromosome. Look at that chromatid over there. Next thing that happens, the nuclear envelope breaks down. Another thing people often say is that the nucleolus disappears at this point, but the nucleolus is just a dense region of chromatin, and when it's wound up, when all the chromatin is wound up as chromosomes, it's all dense, so that you just don't have a nucleolus. So the nucleolus is just an overlapping region of the chromosomes that have all the genes associated with making ribosomes. So that's gone for the time being. We're not using any of our DNA for right now because it's bound up too tight. So we packed up our bags and we got the door open. We broke down the nuclear envelope. You gotta get out of your house if you're going on vacation, right? And next, you gotta get the car ready. So what does that mean? That means that these centrioles are gonna start migrating to opposite poles. These are our travel arrangements. And behind them, they start unspooling these microtubule fibers. And this is the this is the mitotic spindle. So centrioles migrate. A lot of stuff happens in prophase. Did I spell chromosomes wrong? Oh, good catch. I need more coffee. Thank you. Yes, metaphase will be next. So the centrioles migrate. Mitotic spindle fibers are going to attach The thing is that I'm fighting with this tablet, so periodically it just doesn't register and it'll skip a letter, and I guess I just didn't notice and I just kept writing the word as though there was nothing there. Mitotic spindle attaches to the centromere. What's a centromere? I didn't emphasize it too much, but it's just the waist right here. It's the middle part of the chromatid where the 2 chromatids are bound together. Centromere is attached to the chromosomes. Centrosome is a network of proteins around these centrioles. Okay, so the mitotic spindle comes from the centrosome, but it attaches to the centromere. So, yeah, unfortunately in biology, we frequently use, we'll have like 12 terms that are off by like one letter or two letters, and they mean entirely different things, but they're all in the same like subject area. So you just have to sort of repeat these things over and over until it starts, until it's just locked in. Okay, so prophase, P, we have the packing phase. Metaphase, middle. I call it the middle phase because, two reasons. The chromosomes line up in the middle because these spindle fibers, these centrosomes, are pulling on them in opposite directions. It's like tug of war. Right? And if you have really evenly matched teams in tug of war, what happens to that little ribbon you tie in the center? It goes right in the middle, right? It doesn't budge too much left or right. So that's what's happening. They're lining up in the center, and they line up on this imaginary line, which we call the metaphase plate. Plate because this is a three-dimensional object. It's a sphere, so if we draw a line through a three-dimensional object, we are drawing a circle, right? So that's the metaphase plate right there. So chromosomes line up. In the center on the metaphase plate, which is essentially the prime meridian of the cell there. The other reason why I call it the middle phase, get rid of my little diagram here. The other reason why I call it the middle phase is that there's two more phases to go. Ooh. Might need more space for myself. I'll do it like this. Two more phases to go, and it was preceded by interphase and prophase. So it is literally in the middle. If you write down all the phases like interphase, prophase, metaphase, anaphase, telophase, it's in the very center of that chain. Now that would be ignoring for a moment that interphase is made up of three subphases, but you know, it's a mnemonic. So the chromosomes are lined up in the center in metaphase, but in anaphase, we put so much tension on these chromosomes that they snap. They break at the centromere, and I have that arranged, yep. They break at this centromere here, and the sister chromatids go to opposite poles. Yes, in metaphase, just regular old metaphase, the sister chromatids separate from each other. And now they're no longer sister chromatids. Now they're what we call daughter chromosomes. Once they're separated from each other, each chromatid is a chromosome in its own right, and that's how you can go from having a parent cell with 46 chromosomes to having two daughter cells with each 46 chromosomes, because we break the sister chromatids in half, and then during the next S phase, we duplicate those chromatids again. So that is anaphase. sister chromatids. Oh, I should be consistent wherever I can. Sister chromatids break apart and become no sense in writing it twice. Become daughter chromosomes. And they migrate to opposite poles. And once they get to these opposite poles, they can start loosening up again. And we start rebuilding the nuclear envelope. Now we have two daughter nuclei. Yep, this is the cleavage furrow is coming up now. So anaphase is the away phase. It's where we pull the two sister chromatids away from each other. Then in telophase, these two daughter nuclei, well, it's kind of sad. They were sister chromatids, then became daughter chromosomes, and now they're daughter nuclei. They're never going to see each other again. They're going to be in these separate cells for the rest of their life, but maybe they can telephone each other because we're entering telophase here. Now, in telophase, we're going to undo everything that happened in prophase. So in prophase, the chromosomes condense. In telophase, it returns to loose chromosome, right? We have chromosome. We have loose chromatin. Loose chromatin, so we went from condensed chromosomes back to loose chromatin. We rebuild the nuclear envelope. The mitotic spindle began forming in prophase. Here, the mitotic spindle breaks down. And generally, we're just sort of returning to the way things were during interphase here. So we're kind of going back to that point, but we still have two daughter nuclei inside of a single cell. So what's going to happen, you guys remember that the cytoskeleton runs absolutely everywhere, right? Well, there's a ring of contractile proteins, which goes right in between these daughter nuclei. And just like in endocytosis, where we were able to pinch off a little bit of that membrane to make a vesicle, here we're going to pinch the entire cell in half in just the same way. So when this gets smaller and smaller and smaller, eventually these two portions of the membrane are going to hit each other in the center. They're going to kiss, and then these will be pinched apart. Now that, because this is a liquid membrane, so it's like it's made out of a fabric almost, as we continue to pinch it and pinch it, you start getting these little wrinkles coming out. So if you look at it under a microscope, under like an electron, a scanning electron microscope, you'll see little wrinkle patterns starting to come in. So we called this the cutting wrinkle, but we like to couch things in more flowery language than that. Instead of the cutting wrinkle, it's the cleavage furrow. The cleavage furrow is what is actually going to pinch it in half. And then, not another phase, but we have something else is going to happen called cytokinesis. Let me get a little bit bigger, give myself some room. Cytokinesis means cell movement. Cyto means cell, kinesis means to move. So we're moving these two cells away from each other. Let me get this so it's the same size as everything else. Great strength and weakness of mine is that I think things should look good. But sometimes that means I just spend too much time on little details. So in cytokinesis, ooh, why did that not work? Oh, because it's still selected. In cytokinesis, we take these two halves and we pinch them apart, and then we have daughter nuclei and daughter cells. Two diploid, because they all have 46, they all have the full set of chromosomes, right? We started out with 4 chromosomes here, and then 4 chromosomes went to each side. So each of these daughter cells now has 4 chromosomes. 2 diploid identical daughter cells. Okay. 2N, if we're going to get the abbreviation in there as well. And now each of these daughter cells is back in G1 of interphase. Okay. Oh, that one's just my fault then. Can't blame the tablet on that one. That one was me. All right. So that is the steps of mitosis going through. You have G1, S, G2, followed by, what's the issue here? There we go. Followed by prophase, metaphase, anaphase, telophase. Cytokinesis is when the cleavage furrow breaks these two bad boys in half, these two bad girls in half, I guess, their daughter cells. And then each one of these is back in G1 to start the cycle over again. And it'll go through 90% of its life before it gets ready to do mitosis one more time. So, slight difference for plant cells, because plant cells have a cell wall. So before they're able to pinch themselves into, oh, hi Edward, bumping my arm. Before they're able to pinch themselves into, they have to build a new section of that cell wall, which we call the cell plate. It's located pretty much where the metaphase plate would be. So in the case of plant cells, there actually is a literal plate that we're going to place in the center. And then once that plate is built up, we can get these two cells separated all the way. Okay. So cytokinesis is a little bit different in plant cells and animal cells because plant cells have this cell wall to contend with.